Hello, everyone. My name's Paul Denny. I'm one of the curators at UniProt, and my intention today is to give you a kind of introduction to what you can do with the UniProt resource, in particular, using the website, how you can look for your proteins of interest, find out functional information about those proteins, their sequences, specific features of those proteins, how those proteins fit into a proteome. So a proteome is the collection of all the proteins encoded by a genome. And it's one of the ways in which we share our data. And then say a little bit about how we also curate mutation and disease data. And how those proteins are implicated in disease. So what is UniProt? It's a universal protein resource. It was produced originally by a consortium, a collaboration between 3 big bioinformatics databases at institutes in the UK, in Switzerland, and in the US. And I am just going to remind myself to turn on my laser pointer. Hopefully you can see that now. Yeah, so these are the logos of those 3 resources, the 3 institutes. And UniProt consists of a freely available, very comprehensive collection of sequence and functional information. It includes the primary sequences for isoforms of those proteins. We focus entirely, with a few exceptions, on the physiological function of those proteins. So where are they located in the cell, the pathways that they participate in, where they're involved in some kind of chemical reaction, biochemical reaction, their interactions with other proteins, how they may be involved in disease, and then structural information. And I'll say a little bit about that later. And then linked up with all that data are various analysis tools such as BLAST and also multiple sequence alignment. So our website is organized in such a way that it allows a user to download data. You can choose the format you want that data in. It helps with facilitating visualization of the data. There's a very powerful suite of search tools to help you find the information directly from the website or in programmatic ways. So you can do batch retrieval and also more powerful and comprehensive searches using, if you've got the right skills in coding, using the API. You can customize the way that data looks when you download it, and we supply ways to explore the protein data and then analyze it. So Ensembl gets its data from a variety of sources. Sorry, not Ensembl, UniProt. I was misled by my own slide. There was a picture of the Ensembl logo up here. The vast majority of the data comes from the international protein sequence databases, INSDC. So that's a consortium of 3 databases, again in the US, but in this case in Japan and Europe. About 95% of the protein sequences come from there. But then we also draw down information from Ensembl, from PDB, from the structure database, and we get direct submissions from some people who send their data directly to us, to UniProt. This is what you see on the homepage when you go to our website. It includes various things that I've highlighted. So in green up here in the top left, there are some of the tools that are associated with analyzing protein sequence data or obtaining specific sets of protein data. And then in this purple blue shade at the top here on the right, this tells you which version of our data set is currently displayed on the website. So we've got release 2025-03 at the moment, and the next release is being prepared at the moment. So there will be some changes in the months ahead. Then down the bottom here, where it says resources, we present our data in various different contexts. So you can think of it as the knowledge base. So this is all the proteins in the database. Or you can think of it per species. I've mentioned proteomes earlier. So this is a way of obtaining all the proteins encoded by a genome. Then there's also different clusters of data, proteins which are clustered together based on their degree of sequence similarity, sequence alignment. And you can obtain these as a complete data set at different thresholds of similarity. And then there's a bigger database which underpins all of this called the Sequence Archive, or UniProt. And this includes other sequence data, which has, I'll say a little bit about later. Now, in the middle of that view of the website, you could see a search bar, and I've zoomed in on it here. You can see a little bit more of the kind of detail. The search bar allows you to type any free text in here, a gene name, a protein name, a disease, whatever you want. But then if you want to be more fine-grained in your search, there's also an advanced search button just here. And let's say you've got a list of accession numbers or identifiers from another database or from UniProt itself, and you want to look at all of those together. You can do a search using the list facility. I'm going to take you through some of the things you can do with a few examples. So let's say you've got an interest in the biological process of autophagy or autophagy. And as many of you probably know, this is the process whereby cells recycle their protein contents. And it can be stimulated in various ways. And so I've picked here some proteins. These are their gene names, protein names, ATG12, 5, and 16L1. And I've shown them in a very simplified, highly cartoon style pathway. And you could choose to put these individually into a search bar, or you could use the advanced search. And so if we take this example, let's say we're going to do an advanced search. You could choose to restrict the search by taxonomy, and then you could type in a gene name here, and then you can use Boolean algebra, Boolean expressions to combine different search terms. And you can add more by clicking on add or remove, and choose extra criteria for restricting or expanding your search of interest. One other tool I mentioned earlier was the ID mapping tool. And this allows you to take a set of identifiers or protein names and search for them all at once. So in this case, I've taken those three identifiers I mentioned earlier, ATG5, 12, and in this case, 16, and then put them into this text box. And then I can choose to tell the website that these gene names are, well, these identifiers are gene names, and I want to get accession numbers from UniProt for them. And I could choose to then restrict this by species. In this case, I've decided human. And then the website will go away and perform that. And one of the things in this results page that you can see here, all these searches are saved for 7 days, and you can return to them at any time. You can go back to the website and find the results. Now, the results of this kind of simple search should be over in a few seconds. But if you add a large number of identifiers, it may take longer. So, and in some cases, you may want to keep the results for longer than 7 days. And you can do that using one of the options on the search tool facility. So let's say we've done that search, and in this case, there's a deliberate mistake. I've accidentally, when I was doing this, typed in, instead of ATG 16, I've typed in ATG 161 or 161, and it tells me immediately in the results that this is not mapped. There is no ATG 161 that matches this in the human proteome. So there's no human proteins that match this. But I have got some results back hidden under here. We've got various types of result for ATG5. And I'll say a little bit more about the different types of result that we've got here in a moment. So one thing you might have noticed in this box on the left here, there's two classes. There's a gold icon here, which refers to reviewed entries, and a silver one here, which is unreviewed. And I'll say a little bit about those in the next slide. So the knowledge base that UniProt maintains can be split up in two ways, or one way of dividing it up. There are entries which have been reviewed by curators manually. And so all this sequence information, or much of it rather, has been abstracted from scientific publications by expert curators. And we've reviewed those. And we've also done things like grouping together isoforms, all derived from one gene of a particular species. They've been grouped together and included in an entry, and that then becomes a reviewed or SwissProt entry. And underpinning that, or our starting point, are the unreviewed entries. And these have been obtained from direct sequence submissions. So they come from the databases that I've mentioned, from those sequence repositories. They've been analyzed, they've been annotated and analyzed computationally. So they've been through automatic annotation pipelines created at UniProt. And one difference between the two data sets, other than the fact that the gold star set, the reviewed set, has been checked manually by curators. One difference is that protein isoforms derived from the same gene for a particular species are not separated out, are separated out. They're not grouped together in the unreviewed set, or the tremble set as we refer to it. Going back to our hypothetical advanced search using a set of identifiers. In this case, I've imagined an alternative scenario where instead of having protein names or gene names, we've actually got the UniProt identifiers. Let's say we found them in a paper somewhere, or some other resource online. And we want to know what these identifiers, these UniProt identifiers, because that's what they are, what their corresponding genes are, and which protein entries they represent. And so what we could do is to go back to that ID mapping tool, type in those identifiers, and then we could tell the website that we know these are UniProt identifiers, where it says UniProtKB ID. And then we say, well, we'd like to know what gene names these are derived from. And although I've neglected to include this here, oh no, actually it's here highlighted in green. This result is what you obtain. So for example, 094817 is from a gene called ATG12. Another tool that is shown at the top of every web page, the home page, is called a peptide search. And this is useful if you've got a small to moderate size, up to 100 sequences of peptides from, for example, if you work in a lab that creates proteomic data, you might have a small or even a larger number of peptide sequences, and you want to know which proteins do these sequences represent, what do they come from? And in this way, you can do this with up to 100 different peptides. And in this particular example that I haven't given you the peptide sequences, but you can see that we've got, if we did a peptide, this particular peptide search, it corresponds to 2 reviewed entries in SwissProt and 1 unreviewed one in the Tremble part of the knowledge base. Taking a pause there for a moment, if we move away from the sequence data, once you've got a result from a search and you want to find out more about the function of a particular protein, however you get to it from the website. In this example, I'm going to just talk you through the kind of functional information that we store in the UniProt database. So this is an example of a protein that I've picked that's involved in the immune system, amongst other things, in an inflammation. And it's a subunit of a complex. It's involved in inhibiting the NF-kappa B signaling pathway. It's quite a key protein in the human immune system. So down the left-hand side here, I've highlighted all the kinds of types of information that UniProt stores for a protein, and it's highly various. There's taxonomic information, the location of the protein within the cell, any role in disease, and perhaps if there are any known sequence variants for that protein, either natural variants or those that have been created in mutagenesis experiments, post-translational modifications the protein undergoes, expression data, and so on. So this particular this particular protein is an enzyme, and this is one of the sets of data that we put a lot of effort into capturing from the literature and also through our automatic pipelines. And so catalytic activity is a key part of the data that we retrieve and curate for proteins of interest. We have links to many external resources, especially for things like enzymes. So what we'll have is an EC number, the enzyme commission number here, links to the RIA database, which maintains the details of chemical reactions that an enzyme catalyzes. And then in this example, I've included a screenshot of some of the different participants in a specific reaction that this enzyme catalyzes. I mentioned subcellular localization earlier on. This is one of the images that we use to present a visual view of the subcellular localization of a protein. And I do believe this was captured for that last example. And so this protein moves backwards and forwards between the cytoplasm and the nucleus. And that's why it's been localized to 2 locations in this entry. And this kind of yellowy color is meant to show that that's what the protein does. It's found in both the nucleus here in the middle of the cell, and it's also found in the cytoplasm. Underpinning so much of our data is the literature. So the manually curated data is almost all derived from the scientific literature. And in this particular example, I've highlighted the information that we display. So the abstract, authors, title, and then links to PubMed and Europe PMC for the full text, where it's available. And then the publication detail, the name of the journal and when it was published. And then highlighted in green here in this box, this is quite an important part of the the way that we show literature citations on the website. What this does is it shows to you or to any user, where has this paper been cited on the UniProt website? And was that citation created manually, or was it mapped automatically? So this little icon here is meant to show an automatic mapping that's been created by one of the automatic pipelines. And the number here is the number of different entries which map to this particular publication. And this number could be a lot larger. I should have actually chosen a different example with a number slightly higher than 1. One of the other great joys of, in my opinion, of the UniProt website is the richness of cross-references to other people's data, as well as the manually curated data created by the UniProt curators. We have links to over 180 different external databases, and they supply a lot of different information of interest to users. And so one of the databases that I was involved in trying to get, not trying, actually we successfully got cross-references to, was one that specializes in antimicrobial resistance, and that was one that was added recently. Earlier on, I mentioned about the sequence data details and the analysis tools that entries have associated with them. So in this example now, I'm going to step you through some of the kinds of information you can find for protein sequences, and also the tools you can use to analyze those sequences. This is the kind of thing you'd see on a protein entry, a UniProt entry. This is just one version. You can download it. We go up here. You can see it says download. You can download this information in a range of different formats. But if we go back to a page on the website, one version for every entry of the view of a protein entry. You can see across the top here, we've got tabs where it says entry, variant viewer, feature viewer, and that's the one which is, there's a thin blue line underneath there. That shows that that is the feature viewer view for this particular entry. And down the left-hand side here, these are the different types of annotations to the sequence that we present. Things like domains in a protein, and I'm going to say a little bit about one of those in a moment for this particular entry. But then there are things like active sites, sites of post-translational modification, how much of the protein sequence is represented in a structure. So in this example, this green bar here shows you that quite a large part of this sequence has been captured in a structure. And then one other thing which is of great interest to many users is that because we have links with the predicted structures of proteins from the AlphaFold database, and also the predicted effects of variants in the alpha missense database. Across the bottom here, we've got different levels of confidence in the predicted structure in AlphaFold. I said I was going to show you the kind of thing that you can do with that previous view. If we had clicked on just over here on the right-hand end of the protein, the far end, across the top here, we've got a sequence bar. So you can see the protein is 745 amino acids. In fact, it's written at the top here in this view of the entry. Near the end, near the C terminus of the protein, there's a specific domain or region, which if this was a live version of the website, I could click on that and show you it. And this tells you that this particular part of the protein is known for binding to other proteins called NEMO. And then at the same time, what that does is it highlights that part of the protein in the structure. So in the bottom right-hand corner here, we've got highlighted in green in the PDB-derived structure. This corresponds, in green, this corresponds to this region in the entry viewer. Moving on from the feature viewer, I mentioned being able to do BLAST. Well, we could go back to here. Actually, no, it's not viewable there. In the entry view, you can see that's what this is because it's the little blue underlining here for entry. On the left-hand end of this collection of links here is something called Tools. And then when you click on this, there's a drop-down menu, and one of the options is BLAST. So you can find proteins that are similar to this simply by clicking on this link here. And what will happen is that the sequence, the main isoform, the primary isoform, the one that's displayed on the website for that entry, is then automatically pasted for you into the search box so that you can do a BLAST. Or alternatively, you could come to this page with your own accession number. If you've got a protein accession, say a UniProt ID, you could paste it in here, or you could paste a sequence into this dialog box. And it's quite flexible. You can choose to which specific part of the UniProt database you want to look at. Do you want to only look at the reviewed entries, or perhaps you want to include all the reference proteomes and the reviewed entries? There are a lot of different options from this drop-down menu. And then you can restrict that search by taxonomy. And as an example, using that protein that we saw earlier, we can see there's a whole range of proteins that have a hit in this BLAST search. 5 of them are reviewed entries, and there's quite a large number of unreviewed ones. And over here on the right, this bar here shows you the extent of coverage. And then if you click on the link, you can see just how similar your proteins are. So in this example, I've clicked on a mouse ortholog of this protein, or a homolog. And at the bottom here, you can see the alignment of the mouse and human sequence. And you can see there's a lot of similarity. And you can customize the way this appears on the screen. You can change the way it's shown. At the moment, this is just the first few amino acids in the alignment. And if you click on wrapped here, what happens is you then see the whole of the alignment, and you can scroll down and see more details. The other thing you can do is you can choose to, perhaps you want to see more details in the alignment of multiple hits. So what you can do is, in these click boxes on the left here, you can choose to select some of the hits and then do a multiple sequence alignment with them. So in this example, we've just selected a few of the hits and align them using the multiple sequence alignment tool that's built into the UniProt website. And there are various ways that one can do this. It doesn't have to be the results of a BLAST search. You can come into the align tool with a set of IDs, identifiers, paste them in up here in this first dialog box, or you can paste a sequence in, and you can choose multiple sequences. They have to be in FASTA format. As you can see, this has got the header and then the sequence underneath, following the FASTA format. An example of the kind of thing that could be returned, this is that previous protein, and it's been aligned with, there are 3 proteins, human, mouse, and cow. And they're very highly conserved. one thing that you can choose to do with this result, you can look at the annotations from one of the proteins in the context of the whole alignment. So let's say I'm interested in the active site, and I know the active site has been annotated in one of the entries, in this particular case in mouse. And so what I've got here is I've highlighted the active site, and this dot here this is a key residue in the active site for this particular enzyme. And as you can see, it's conserved between the three orthologs from the three different species. Can't seem to always get the slide to advance. There we go. Another example now, looking at one other way in which we present the data within the UniProt website. I mentioned earlier on proteomes, and that this is essentially, it's all the proteins encoded by the genome for a particular organism. Now, our data set is extremely rich. It represents over 160,000 proteomes from across all the super kingdoms. And almost always these are derived from completely sequenced genomes. Each proteome has a unique identifier, and there's all kinds of information for those species also built into the UniProt website. I mentioned disease and mutation data, or mutagenesis data. One part of the UniProt web page, the home page, if you scroll down, and that's what we're showing here, is this section called supporting data. It allows you to go straight into looking at specific human diseases. And I'm going to say a little bit about some of those now. So in this example, I've chosen Parkinson's disease. And down the left-hand side here, you can see it says disease and variants. So Parkinson's disease is a very complex disease, but there are some types of Parkinson's disease which have a clear genetic predisposition. And so, for example, some of the early onset versions of Parkinson's, earlier onset variants of Parkinson's, have a strong genetic component. And in this example, I've just displayed a couple of subtypes of Parkinson's, known as PARK and PARK2, PARK2. And some examples of the kinds of natural sequence variants that are found in these conditions. Some are definitively shown to be causative, others are just associated. But one other type of not disease, but phenotype information that we store in the UniProt database, when we're talking about model organisms, instead of talking about disease, we usually refer to phenotypes. And so in many protein entries, there will be phenotypic information primarily for null mutants. So in that case, where a gene has been completely inactivated. But there are other classes of data we capture because widely used in many model organisms are techniques that either just knock down the function of a gene or perhaps introduce a point mutation. And so nevertheless, there's a phenotype associated with those, and they're captured in a different part of an entry. So in this example, I've got a disruption phenotype this is a null. I mentioned point mutations. What we'll do is we will, as curators, will capture this kind of information from the published literature, and if there is a mutant, if the mutant has a specific name, that will be annotated. And then we also capture things like the specific amino acids that are affected. So I've given you a whistle-stop tour through the kinds of things that one can do with the UniProt website. I've shown you how to do a search, how you can access some of the functional information for the proteins in those searches and the sequences for those proteins, and how to analyze them. I've said a little bit about exploring proteomes and how you can get disease and mutagenesis data. We aim to get updates to the website data released every 8 weeks. That varies a bit. It's at the moment, I think we are due an update towards the end of the year. And then it will become public early next year, I believe. And you can download the data in a range of different formats. One thing that we always want is we want advice from our users. We want you to give us your feedback on ways we can improve the website and the way the data is presented. So I've just highlighted this here. There's a little hidden in this ellipsoid here. There's a help link, so you can give your feedback there or down here on every page. And if you need help, as well as saying, well, you know, we could present this better, we've missed this information, you can also find help by going to the help center. And there's also more information on things like other training guides and documentations on the website. There's a lot of resources built into the UniProt website to help users. Of course, the other thing is that you can get help from the training team, and this just highlights some of those things. Before I finish up, I need to point out, as I said right back at the beginning, the UniProt consortium consists of 3 very important partners. This shows you some of the members of staff and the PIs involved in creating the consortium. And I also have to say something about our funding. We get funding primarily from NIH, but we also get money from EMBL, from the Open Targets database here at the EBI, and also from the Swiss government. And at that point, I will stop and say thank you very much for your attention.
Thank you very much, Paul. It was a very clear presentation. So probably you didn't notice as you were presenting, but we had around 20 questions coming in Q&A. And David and Adura were very active and replied to basically all of them. But if there are any more questions at this time, please type them now and we can take them live. I'll just take a moment. I see there is someone asking, can we get the slides, please? So yes, the speakers will share the slides, and tomorrow you will find them on the EMBL-EBI training website, on the webpage of the webinar, together with the recording. If you have attended the webinar as well as registered, we will just send an email with the link to those, so it will be even easier to retrieve them. Let me just check the Q&A. So we are getting quite a few thank you messages. While we wait to see if someone is asking any other question, I'd like to ask David or Adura if they have anything else they would like to add to the answer they typed down to previous question. You can switch on your camera if you'd like.
We had a lot of questions come in about the peptide search tool. I'm just going to check one of them because there's one that I couldn't answer completely. And maybe, Paul, you might have an answer to that if I can find it. Here we go. So the question was from Homen Fukan. for peptide search, how can we search for protein match with our peptide from proteomics data from a new organism which does not have any reference in UniProt database? So I kind of understood that as in not being in UniProtKB. I wasn't sure whether this was the case, Paul. If you put a peptide search in, does it return a UniPark entry if it can't find Swiss prot or Tremble entry?
I have to say, I'm not sure, but I think it does. So, for example, I know that if you do a sequence search or even a text search, sometimes what will happen is if you do a search from the homepage, it will sometimes say you can't find it in the knowledge base. Do you mean, and it'll give you a link to the relevant entry in UniPark in the archive. So it might be, this is speculation, I'd have to check. It might be that the same thing would work with a peptide search. So if it was for an organism that currently isn't in our data set in the main knowledge base, but it might be in the archive. It should be in the archive.
I think if it's in the archive, it should definitely find it. But for any sequences that aren't in the archive, it's not going to find it, of course. But it'll give you the closest sequence match that it can find.
Yes. Yeah. We'd need some examples. That would be good. If someone wants us to try, they could send us a few peptides and then try it out for them.
Thank you. We had a few more questions coming in, so we can take them now. Which evidence tags correlate best with prediction accuracy?
Prediction accuracy.
Accuracy.
Well, I would say probably the ones that have been assigned by a curator, so experimental evidence derived evidence tags. But I don't want to demean or denigrate the other sources of information, because some of them will come from external databases, and so that they should be at least as good as data that we've assigned, evidence tags we've assigned as curators. And the same with the automatic annotation evidence. Those are also very accurate, and they are created using manually curated data in the first instance. Those automatically assigned evidence tags derive from a manually curated template.
Just to add on top of that, so to be clear, when it comes to predictions, the curator tags are the mod and the mix tags. You'd have to dig out the numbers, but those two tags refer to functional data that is purely predicted. If it's got an experimental tag on it, then it's not predicted. It's been proven or shown experimentally. So just to clarify the differences between those.
Yeah.
We have then another question, which is how to predict active sites. So it's a very broad question, I believe.
Well, that is done at some level in the rules for the automatic annotation pipelines. Some of them do assign active sites, but they don't predict them de novo. They're based again on some template. I don't think presented with a sequence which has no similarity around the active site, it wouldn't to a known protein. I don't think UniProt would be useful in predicting an active site purely based on that kind of information. It would need to have a template of some kind from a protein of known enzymatic activity. And does that sound sensible?
Can I add a little bit to that?
Yeah.
So, if you have a sequence, for example, and you want to find out if you want to predict the active site, like Paul actually explained, you will need to try to match the sequence with some of the sequence that's already in UniProtKB. and see if they have a sequence match that is very similar, and check that sequence to see if there's a prediction of an active site for that sequence. And you can use that, the alignment tool, to be able to check where on your sequence those active sites correspond to in the sequence that matched your sequence, or that are very similar to your sequence. So you will need, like you said, you will need a template to be able to predict, and that template must have active site already, either from crystal structure or prediction based on and autologs already, before you can be able to predict active site for the sequence you are interested in.
We can move to the following question, which is if you can say something about SparkQL, which I do admit I don't know what it is.
I'm afraid I've never actually used it. I'm embarrassed to say, but I haven't used it. I know that it's an alternative way of querying a programmatic query of the data in UniProt. But other than that, I can't tell you.
It's a way of putting together more complex queries if you're looking to define a group of proteins to a greater degree. Most of us don't use it anymore. We use the advanced search tool, so none of us are experts at using it. But it's there for people who have the ability to use that way to access the database and generate queries of it.
Yeah. You can also, if you click on the SparkQL, you can see there is a documentation at the bottom where it talks about tutorial on using SparkQL with UniProt. So if you need more information, you can actually go there and access that way. I hope that helps.
That's good. Thanks, Adura.
Thank you. I linked this to another question because someone else is asking, please provide more information on API access. So we do have a recording webinar that UniProt ran last year on programmatic access to UniProt using Python. I will now send the link in the chat so you can access it. But I do believe this is a webinar that may run yearly, so more may come this year for another live event where you can have also the opportunity to ask questions. So you can watch the space for more information to come. But if you would like to add something now, please.
Sorry, I was just going to say that there will be an updated version of that webinar coming up in the next few weeks. I think it's planned. I don't know the date, but it's definitely planned.
Perfect. So yeah, I do believe it maybe before Christmas break, so in the next few months. Thank you. So we can take the following question. So someone is asking, UniProt to AlphaFold protein-specific identity number find? Very concise question. So I think they are asking how you match and cross-link the two. So if I'm in one database, how I can understand, oh, this is the UniProt protein.
Probably what it means is if you go to a UniProt entry, you'll find that there is a link to an AlphaFold predicted structure in many cases. I think that's what they're asking.
I was just checking if you could do it in the ID mapping tool, and I only get the PDB mapping. It might be in the ID mapping tool, because it might be looking at, can you map one identifier onto the other? But it doesn't seem to be under a 3D structure database. So that's something I'd have to look into a bit more.
Thank you. So the next question is, how can I search between homologous protein in polyploids, like within wheat or between wheat and other cereals?
Well, I know that wheat is one of the organisms that we definitely have data for, but the distinction between a polyploid and a non-polyploid version of wheat, unless there are sequence differences between I'm not sure if paralogs is the right word. You probably wouldn't call them paralogs. Presumably they are homologs in the duplicated parts of the genome. unless there are sequence differences, we would normally make the assumption that they would represent the same functions. So I know that it gets very complicated in wheat. You've got hexaploid and other degrees of duplication of the whole genome. I'm not sure if that answers the question.
I think the best place to look would be at the end of an entry. So there's two sections called sequence and isoforms, and then there's similar proteins. And under similar proteins, you'll have a list of any entries, usually in Tremble, that have been computationally mapped to be similar to this entry, usually from the same species, but also sometimes across species to identify orthologs and paralogs. Most of these haven't been curated, but the links are there. So if someone would want to identify those sequences, I think that's probably the easiest place to do it.
Yeah, that's a good suggestion.
Thank you. Oh, anything else? Or we can move on to the next one? Okay. So the next question is about the reviewed label. So does it mean that the full length protein sequence and the structure is already confirmed?
Not necessarily.
If I see it.
Many will be reviewed, but won't have an experimentally determined structure. They might have an AlphaFold predicted structure. The protein typically will be full length, but in some cases it's not. The majority will be. But you can tell that from the entry. Somewhere in the entry, it will mention the word fragment, which means that although you've got this much sequence, it's likely or predicted that it doesn't necessarily represent the full-length protein. But nevertheless, because there's experimental data for it, there's perhaps been publications, that's considered useful information for a user. So there are some entries like that.
Thank you, Paul. The next question is about structural alignment. Is it possible to perform it using UniProt?
If by structural alignment you mean not a sequence search, but a direct comparison of the structure of the shape of the structure, the short answer is no. There are ways to do that, I believe, in the AlphaFold database. I think, again, this is not something that I've used very much, but I believe there might be a way to search for similar structures, or if not, possibly in PDB. Did Dave or Adura know about that?
PDB is your best option. What we do have, though, is in the variant viewer, you can align our annotations to, so for features that we have annotated, you can align that to a structure that's linked, that's cross-referenced in an entry. But we don't do structural alignments at UniProt, no. PDB is your best bet.
Yeah. Also, what I know is that if you want to align structures, you probably need to have them maybe on PyMod, and that is something that you have to take the structure out yourself and see if they align. That's normally what you would do when you want to align structures together. But I don't think you'll be able to do quite a lot, because it requires a lot of computer power to align so many structures.
The other option is there are websites which specialize, that they offer servers to allow sequence structure searches. I have used them in the past. I'd have to dig back in my memory for what they are, but there are servers which allow you to take a sequence and say, well, I've got a structure for this protein. I want to know which other proteins have a similar structure.
I've seen that someone in the Q&A has written, for structural alignment, DALI can be used. So I'm going to send the link in the chat.
That's it. Yeah,
Yes.
DALI is one of them.
Yeah, so I retrieve it and I send the link on the chat in case it can be useful. It's 4, but we have like 2-3 more questions. If you can stay 5 minutes more, we can take those. Okay, thank you so much. Okay. Is there a feature, sorry, or there are plans to incorporate conformational flexibility metrics, like for example, predicted B-factor or ensemble models, either directly in UniProt annotations or via cross-references?
There aren't any plans to do that, but I'm pretty sure we have cross-references to perhaps not conformational flexibility, but I know that we have cross-links to websites that annotate predicted disordered parts of proteins. And we do annotate in the sequence, in the automatic part of the pipelines, there is a prediction of sequence regions which are disordered. And often they correlate with the flexibility of a part of a protein. But it's not something that we're planning to go beyond that.
If by B factors you mean the AlphaFold B factors, those are included in the feature viewer and are mapped onto the sequence there. So we have a track on the feature viewer which shows directly the B factor from the AlphaFold prediction, but that is imported through a cross-reference to the AlphaFold database.
Thank you. I do think the last questions that we have is also related. As they say, can we get the similarity check between isoforms of a single gene in a particular species, for example, in Arabidopsis, and show it in the 3D structure?
I think the answer is probably no, because the website is set up to display a primary sequence, a selected isoform for a particular species, for a gene from a particular species. If you wanted to see an alternative view for one of the isoforms from that gene, and then see it in the 3D structure, I think that's beyond what we can offer.